Hello, what is amino acid? Amino acid comes from protein, where it is the monomolecules of protein which divided into exogenous and endogenous. Exogenous protein are based in dietary sources not exist in body, endogenous protein which exist in the body. Example, exogenous is animal while endogenous is enzyme.
Digestion of amino acid. When protein sources get ingestion orally, it will get in stomach. Protein will hydrolyzed by pepsin enzyme into peptides, then into intestine, where the peptides will be hydrolyzed into a smaller peptides by trypsin and chymotrypsin enzyme. Lastly, it convert into amino acid by carboxypeptidases. Absorption of amino acid. Digested amino acid will be absorption into the body through intestinal membrane, either brush border or basolateral. First, through brush border, amino acid will absorb with passive as exchanger or active by transmembrane ions into the membrane. Next, absorption through basolateral membrane, where amino acid get absorbed into extraintestinal tissues. Then, the amino acid will be either enter the portal vein to liver, transport into hepatocytes, or transport into other cells.
Hepatic catabolism and uses of aromatic amino acid, phenylalanine, and tyrosine. Phenylalanine and tyrosine are partially glucogenic because they are degraded to fumarate. Moreover, phenylalanine and tyrosine also catabolize to acetate and thus are partially ketogenic. Degradation of phenylalanine is specific to the liver and the kidneys. First step, phenylalanine is converted to tyrosine by the enzyme phenylalanine hydroxylase, also called monooxygenase. 2. Tyrosine degradation begins with transamination by a vitamin B6-dependent tyrosine aminotransferase to yield p-hydroxyphenylpyruvate. The compound p-hydroxyphenylpyruvate is formed and then it is decarboxylated by a dioxygenase to generate homogenizate. 3. Homogencytase dioxidase converts homogenizate to malonyl acetate, which is the isomerized to fumaryl acetate by isomerase. 4. The fumaryl acetate is converted into fumarate and acetoacetate by hydrolase. Then both fumarate and acetoacetate is metabolized to acetyl-CoA for energy or ketone body production. Overall metabolism of phenylalanine and tyrosine. Other compounds in the body cell also can be synthesized by tyrosine. For example, in neurons and the adrenal medulla, tyrosine helps to synthesis neurotransmitter and hormone. Disorder of phenylalanine and tyrosine metabolism. 1. Phenylketonuria, PKU, occurred when the activity of phenylalanine hydroxylase is defective. 2. Tyrosinemia 2 due to impaired activity of tyrosine aminotransferase. 3. Alkaptonuria due to defective homogenizate dioxygenase activity. Hepatic catabolism and uses of sulfur-containing amino acids. Methionine, a sulfur-containing essential amino acid, is needed to generate another S-containing non-essential amino acid, which are cysteine. The first step of the metabolism is methionine are converted to S-adenosylmethionine or SAM with the help of methionine adenylyl transferase. Next, the second step, removal of methyl group from SAM, will produce the compound S-adenosyl homocysteine, SAH. From the compound SA, the third step of the metabolism occurs, which is the formation of homocysteine with the help of enzyme S-adenosyl homocysteine hydrolase. Homocysteine, once formed, can be converted back to methionine either in a vitamin B12 and folate-dependent reaction, or it can be further metabolized to form cysteine. Methionine will be oxidized to yield succinyl-CoA, which makes it categorized under a glucogenic amino acid.
Next is amino acid cysteine and its functions. Cysteine catabolism begin with the oxidation to cysteine sulfonate catalyzed by cysteine dioxygenase, where the final products are taurine and sulfite.
The final products are important in the retina and synthesis of sulfolipid and sulfoprotein. The disorder of cysteine metabolism cause formation of cystinuria.
Catabolism uses of branched amino acids. BCAA. Liver less important in metabolized branched-chain amino acid which are isoleucine, leucine and valine. BCAA mostly taken by skeletal muscles and others. First, BCAA will be converted into branch-chain alpha-keto acids by branch-chain aminotransferase. Branch-chain alpha-keto acids will convert it into R, IB, MV, 4-CoA and convert to either succinyl-CoA or acetyl-CoA. The conversion of branch-chain alpha-keto acids uses enzyme branch-chain alpha-keto acid dehydrogenase complex which can be converted into its inactive form. Waste product are carbon dioxide and NADH.
Hepatic catabolism and uses of basic amino acids. Lysine is categorized as a ketogenic amino acid as when it is catabolized it will produce acetyl-CoA, an essential carbon metabolite involved in TCA cycle. The most common pathway is the saccharopine pathway. Besides that, lysine are also used in carnitine synthesis to oxidize fatty acid. There are two types of disorders when the metabolism pathway is disturbed, firstly glutaric aciduria type 1 and alpha-ketoadipic aciduria. Arginine is categorized as a glucogenic amino acid as it generates alpha-ketoglutarate. This amino acid has several of its uses when it is catabolized in the liver, brain, kidney and intestine. Histidine is also a glucogenic amino acid yielding alpha-ketoglutarate when degraded. In the pathway, histidine is first converted to urocanate by histidase and with the help of few other enzymes, it is further metabolized to form glutamate. However, this amino acid is also used as a precursor to histamine and carnosine biosynthesis. Hepatic catabolism and uses of other selected amino acids. Let's start with threonine and its function. The difference between glucogenic and ketogenic amino acids. Threonine is metabolized by three different pathways and the common pathway is cytosolic threonine dehydratase. The other pathways are threonine cleavage complex and threonine dehydrogenase where acetyl-CoA and pyruvate are produced. The disorders are propionic and methylmalonic acidemia.
Next is amino acid glycine. Glycine metabolism is done by glycine cleavage complex that produce 5,10-methylene tetrahydroflate, carbon dioxide and ammonia. Disorder of glycine metabolism cause non-ketotic hyperglycemia.